chapter 6, introduction to viruses, viroids, and prions. Okay. The search for the elusive virus. We didn't know that viruses existed until actually just pretty recently, in the last 100 years. Louis Pasteur postulated that rabies was caused by a living thing that was smaller than a bacteria. Okay, that's back in the 1880s. Ivansky first proposed the term virus. Virus means poison in Latin. That's where the term virus comes from. Okay, Ivantes and Bergerac showed a disease in tobacco that was caused by a virus back in the 1890s. So not that long ago, we had no clue what a virus was. Okay, 1950s, virology was a multifaceted discipline. We realized that all these little things cause all kinds of problems. Okay, viruses. Okay? Viruses are non-cellular particles with a definite shape, size, and chemical composition. The big key here is non-cellular. Viruses are not cells. They do not have the mechanism to produce energy, to produce proteins, to replicate itself. It has to all depend upon the host that it infects. So they're non-cellular, but they do have a definite size, shape, and chemical composition, which we use to distinguish them. Okay, there's no universal agreement on how and viruses originated. Okay, viruses are considered the most abundant microbes on Earth. They played a role in the evolution of bacteria, archaea, and eukarya. So they play a role in the evolution of everything, basically. They're obligate intracellular parasites, which means they have to, okay, infect a host, and that's how they reproduce and function. They cannot function on their own. Okay? So properties of the viruses are the obligate intracellular parasite. They have to infect bacteria, protozoa, fungi, algae, plants, or animals. They have to infect a cell to be able to function. They're ultra microscopic. These are very, very tiny. 20 nanometers up to 450 nanometers. Roughly the average is about 50 nanometers, like the COVID virus. It's roughly 50 nanometers inside. Nanometer is 10 to the minus 9 meters. So 0.00090 zeros, and then a 1. Really small guys. They're not cellular in nature, which they have to infect the cell. The structure is very compact and very economical. They package their DNA very efficiently, or RNA. Okay, they do not fulfill the characteristics of life. They're not considered alive because they cannot replicate on their own. Okay, viruses are, you could spend years studying different viruses. They're amazing what they can do. And you're just gonna touch the surface with this class, but if you get into it with virology, it's tremendous what these things are capable of. Inactive macromolecules outside the host cell, and they're active only inside the host cell. Okay? The basic structure consists of a protein shell. We call that a capsid. That is the protein shell, and it surrounds a nucleic acid core. The nucleic acid core can be DNA. The DNA could be single-stranded or double-stranded. It could be RNA. The RNA could be single-stranded or double-stranded. Okay? It cannot contain both DNA and RNA. It has to be one or the other. So nucleic acid of the viral genome is either DNA or RNA, but not both. Okay? That will be on your test, on your midterm. I remember putting that question on there. So remember that one. Cannot have both DNA and RNA inside the capsid, inside the virus. It has to be one or the other. And they can, like I said, DNA can be single-stranded or double-stranded. RNA can be single-stranded or double-stranded. Okay, so the nucleic acid can be double-stranded DNA or single-stranded DNA, single-stranded RNA or double-stranded RNA. But they can't mix. It can't be both. So the molecules on the viral surface impart a high specificity for attachment to the host cell. That's what we use when we make vaccines. In other words, proteins on the edge of this virus. These proteins is very specific, binding to cells and identifying the virus. So how like the influenza vaccine works is we take the CDC, Center for Disease, picks 20 viruses we think we're going to be exposed to, and they guess. And they say, these are the 20 that are going to be important this year. And we take those viruses and we remove the DNA and the RNA. Okay, so they're what's called attenuated. It cannot cause infection. And all it has is a protein coats on the outside. And we incubate that in an egg, okay? And we inject that into people. And the protein coats on the outside triggers a response from our immune system because it recognizes it's foreign. So we build up antibodies to this protein or to this receptor on the virus. Okay? So when the real virus comes along, 3-4 months down the line, we have these antibodies in our system and they respond to it very quickly. We don't have to build up an army of antibodies and they kill the virus. That's how vaccinations work. So you can never, when people say they get the flu vaccine and it causes them to get sick, that's impossible. There is no RNA, they're attenuated, they're dead. There's no way that virus can multiply in your body.
Sorry to interrupt.
All it does is act. Sure.
So, like you said, when people say, oh, you know, I got the flu vaccine and I got the flu from it, that's impossible because it's lacking RNA or DNA, because it gets removed once you incubate it or whatever.
And it's attenuated.
Okay.
Now, can you get the flu even though you got the vaccine? Yeah. You could have got a different strain than the 20 they picked. So you can still get sick. They just picked the 20 most common and they guess, and that's what they do. So you can still get sick, but the flu vaccine itself cannot give you the flu. Anyone that says that is they don't understand what's going on. Cannot happen. Okay. You can have a sore arm, stuff like that, and you can still get sick. You can still get the flu. It's just not one of the 20 that your body's vaccinated against. Does that make sense?
Yes, it does. I always wanted to know how that works, like even with like the COVID vaccine and stuff.
Right. Now the COVID vaccine is different. That went to the market way too fast. That hasn't been tested. We haven't gotten everything out of it yet. So that's why that one does get you sick. Like a lot of times the second injection, a lot of people get sick with that.
Yeah, like
that was,
yeah.
That was not modified yet. That's been on the market way too fast.
Do you
But the
think
regular flu vaccine, I'm sorry, go ahead.
do you think that one still contains the virus RNA or DNA? Because that one I got extremely sick with.
Right. That one you can't get really sick with. That one's not modified yet as the other ones have been with the influenza vaccine. Like I said, we were in a rush to get that out, and that vaccine still has a lot of questions to be answered with that one. Does that make sense?
Yes.
Okay. But I mean, either not counting COVID, all the vaccines, normally deaths in America, about 64,000 die from the flu every year. That's what they've been dying from every year, from right around there. That's just a normal flu. That's not counting COVID or anything like that. So flu vaccines are very worthwhile getting. Okay? The COVID vaccine, I'm not going to enter that debate. Everybody has a different idea of it. Chances are it's a good thing to get, but it went to market way too quick. That there just hasn't been enough time to test it effectively. In my opinion, this was a political response to a medical issue, and that's wrong. Politicians should not make medical decisions. Physicians and research, that should be making medical decisions. But be it as it may, it's the vaccine's out there, people are getting it, and hopefully it works. Time will tell. But that's how the normal vaccines work. That's kind of cool how they do that. Okay, viruses multiply by taking control of the host genetic material. So it uses the host DNA and the enzymes and the machinery in place in the cell to replicate itself. Okay, regulating the synthesis and assembly of new viruses. So the only way a virus can make new viruses is to infect a working cell and have that cell and take over that cell's machinery. Take over the DNA and say, this is what we're going to produce. We're going to produce these enzymes and these proteins They're going to make new virus particles, and we're going to assemble new virus particles. Okay. They lack enzymes for most metabolic processes. They don't have them in a virus. They lack machinery for synthesizing those proteins. They have to get into a cell. If the virus is outside a body, outside an organism, it can't do anything. It's not considered alive. Okay. I mean, virology is probably the most interesting part of microbiology. I mean, I could study those and go to those for months. They're amazing. They're ultra microscopic. Most are less than 0.2 microns, so they're roughly 50 nanometers is the size of the virus, which is roughly the size of the coronavirus too. So you gotta remember, they're really, really tiny. So when you get that debate with mask versus no mask and stuff like that, the average mask, I mean, a medical mask has openings that are 300 nanometers. That's the cloth mask that you wear. Okay, so the opening is 300 nanometers, the virus is 50 nanometers.
So we go through the mask then.
Very easily. Yep. Yeah, that's a whole new debate too. I mean, I mean, they say they get trapped by the mask and it's better than not having a mask. So, like I said, this is a political response to a medical issue. Things haven't been developed right. Okay, but those are the numbers that exist. I'll let you make your own decision. The largest viruses average about 500 to 1,000 nanometers, 20 to 50 times in the average virus. Like I said, coronavirus around 50 nanometers, and your average is about 20. But there are some really big ones, like the megaviruses and the pandaviruses. Those are huge viruses. They're 10 times larger than the regular viruses. And you can see, remember we talked about a capsid. Capsid is a protein coat. This right here is the capsid. This surrounds your DNA core. This is the DNA inside the virus particle. And this is the capsid, this whole area right here that surrounds. This is your protein coat. And if you look at the capsid real close, you'll see these little finger-like extensions come off here. These are little surface fibrils. These are what we find that have to bind to receptors to get into the cell. Okay. So viruses bear no resemblance to cells. We don't see the stuff we see in the cell, all the Golgi apparatus, the rough endoplasmic reticulum, the nucleus, the lysosomes, the membranes that we have. We don't see all that in a virus. They don't resemble a cell at all. They're a virus. They're their own special entity. They lack protein synthesizing machinery. They cannot make proteins. They don't have all the rough endoplasmic reticulum, the ribosomes. Okay? They can't build their own stuff. They're incapable of functioning until they get into a cell, and then they take over the DNA of the cell. Okay? So viruses contain only the parts needed to invade and then control a host cell. So those outer particles allow it to bind to a host cell, and then the DNA or the RNA inside the virus is injected in that cell, and now it has something that can take control. Okay? So a virus particle has a covering, which we talked about was the capsid. Okay, that was your protein covering, and an envelope. Envelope is another coming over the capsid, but it is not found in all viruses. Some have an envelope, and some is called naked. Naked viruses do not have an envelope. Envelope ones are enveloped. They all have a central core, though. We talked about the central core of a virus. We said it contains DNA or RNA. That's an or, not an and. It contains one or the other. Now, the DNA is mostly double-stranded, or it can be single-stranded virus. RNA is usually single-stranded, but it can be double-stranded in a virus. Okay. Now, the matrix proteins enzymes are not found in all viruses at all. Okay, so it's not all make it envelopes. They don't have the machinery to make those. Viruses are usually at what's called a crystalline nature. Crystals are repeating units, like salt. If you remember your chemistry, like sodium chloride, magnesium chloride, sodium hydroxide, whenever you have charges, they bind in one charge, and then think of a die, like you roll on the crap tables in Vegas, okay? It has 6 sides and then dead center. So dead center is the positive, and there's 6 points around that die for the negative charges. That's how viruses are. They form crystals by binding to each other and build up this crystalline structure. Okay? It's a regular repeating molecule that gives rise. So it's very orderly. There's one surrounded by six, one surrounded by six, one surrounded by six, and it builds a crystalline structure. Okay? So they form, when they're purified, we form aggregates or crystals just like our inorganic material or salts do in inorganic chemistry. So all viruses have capsids. Every one of them, whether they have a protein coat, an envelope around it or not, they all have capsids. The capsid is a protein coat that surrounds and protects the nucleic acid, the DNA or the RNA. The capsid together with the nucleic acid is called a nucleocapsid. That's a capsid with the nucleic acid. Some viruses have an external coating called the envelope, which we said. Those lacking an envelope are called naked. Though they're naked if they do not have an envelope. If they're not covered, they're enveloped. Okay? Each capsid is made of identical protein subunits called capsomers, each of these subunits. And those are what repeats to give it its crystalline structure. Okay, this is your crystalline structure. This is what general said. This is a capsid. This is a capsid which surrounds your nucleic acid. Okay, this does not have a protein coat around it. I mean, this does not have the envelope around it. Okay, so this is a naked nucleocapsid virus. Okay, this is an enveloped virus. See the protein coat around? This is your envelope. This purple layer right here. This is your envelope that surrounds. Okay, this surrounds the capsule and the nucleic acid. The spikes here stick out from the membranes, and that's what determines its susceptibility to infect different cells. There's two types of capsids, two structural types. There's a helical and there's an icosahedral. The helical is a continued helix of capsids forming a cylindrical nucleic capsid. So it forms a cylinder and it's just a helix, like a spring. The icosahedral is a three-dimensional symmetrical polygon, has 20 sides and 12 evenly spaced corners. So it's built like a soccer ball, which will have these specific repeating polygon units around it. A soccer ball has hexagons. Okay, this is a symmetrical polygon. It has different sides, but there'll be 20 sides and 12 even spaced corners. So it's repeating. So picture soccer ball. That's what it's like. Okay. So the nucleocapsid. Remember, the nucleocapsid is the capsule and the nucleic acid inside. And the helical ones are rod-shaped. They're rod-shaped. They're assembled into a hollow disc. The nucleic acid is put into the center of the disc. And elongation of the nucleocapsid progresses from both ends. Okay, so this nucleic acid is coiled inside as it builds from both ends. That's your helical capsid. Okay, and you can see this as the appearance varies with different types of viruses. Okay, again, 15 nanometers is roughly the average size of a virus. But on this one, you can see it has an envelope. You can see this envelope right around, surrounding the capsid, and then the DNA inside. Arrangements of capsomers vary among isotechnical viruses. This is the icosahedral capsule now. Remember, this is the one that makes repeating polygon patterns. They can vary in numbers of the capsomers with the polygons. And during assembly of the virus, the nucleic acid is packed into the center of the icosahedron, forming a nucleocapsid. This is formed in the center of it. Remember, the other one was basically formed inside the rod shape. And this one is formed inside this repeating units here. Okay, so this is a capsid in repeating units. Okay, see how it forms. And the DNA is the nucleic acid, or RNA is wound around inside the capsomer. Okay, that's what it looks like in real life. This is the viruses here, the particles binding to it, and this is your repeating units building up in the crystal structure. So you can see like a soccer ball. It has these polygons. inside each one. You can see how they're built and arranged. Okay, so you got these polygons and it repeats over and over. Okay. Isosahedral viruses can lack an outer envelope, okay? So they can be naked. The rotavirus is a naked virus. The herpes simplex virus, which we'll talk about in a little bit more detail later on too, is enveloped, has a coating. This is one of the isosahedral viruses. You can see the DNA core. You can see the capsid. This is this blue area right here. This is your capsid, where all viruses have capsids. This is your envelope. This thick area right here. Surrounding it. This is your envelope. It's a coating over the capsid. Okay. The envelope viruses mostly occur in animal viruses. They're acquired when the virus leaves the host cell. When the virus leaves it, this envelope surrounds it to help protect it in the outer environment. Exposed proteins on the outside of the envelope are called spikes. They're there for a sense for the attachment of the virus to another host cell. These spikes that are attached to the outside of the virus are what recognize a host cell and allows it to bind to it and inject its DNA back into the cell to reinfect the same host or infect another host that the host has died from. Okay. The function of both the capsid and envelope, they're different. They both function the same way though. They protect the nucleic acid when the virus is outside of the host cell. Okay. It only stays in the host cell to replicate itself. So the capsid or the envelope or both protect the nucleic acid. It helps the virus bind to a cell surface because of these spikes and assists in the penetration of the viral RNA or DNA into the next suitable host cell. Okay. You can have complex viruses. So not all viruses are alike, just like not all people are alike. Okay. You can have atypical viruses. Pox viruses. Viruses lack a typical capsid. Okay, covered by a dense layer of lipoproteins. Lipoproteins are what helps our cell membranes. So these are a different kind of virus, not a typical virus. Bacteriophages, which we'll talk about those a little bit too. Okay, have a polyhedral nucleocapsid along with a helical tail and then attachment fibers. Okay. Bacteriophages are, we'll go a little bit down the line here too, but bacteriophages are viruses that attack bacteria. That's what the bacteriophages, usually called phages or shortened, just called phages. But those are viruses that attack bacteria. This is a complex virus. This is kind of what they look like. This is a cross section. You can see the nucleic acid inside. You can see the capsid head, which protects it. And this one has a little collar around it. And this collar extends down into these tail pins. That's what adheres to the cell. Okay, these tail fibers bind to the cell, hold it in place. And these tail pins actually puncture the cell wall or the cell membrane and allow the DNA or RNA, nucleic acid, to be introduced into the cell. That's what it looks like, kind of microscopic picture of it. You can see this in the DNA, the nucleic acid material. Okay. Basic types of morphology, they have all kinds of shapes. Complex viruses. You can see how that one, I think the rotavirus has a tail to it. The other ones you can see has this envelope coating over it. Remember, you have the helical ones, which are rod-shaped, and you have the isosahedral one, which are like polygons that stack against each other. And these are both envelope viruses. You see the envelope surrounding the capsid. Okay. The helical is a coil. You can see that right there. And the icosahedral, you can see the DNA more spread out. It forms different shapes. Okay. So complex viruses, like the pox virus, are large DNA viruses. It can be big, and they're flexible. They have a tail with them. They can be flexible to bacteriophage, which means they infect bacterial cells. They attack the cell wall of bacteria. Animal cells don't have a cell wall to attack. Envelope viruses either helical, like the mumps virus or the rhabdovirus, or icosahedral virus, nucleocapsid, like your herpes virus or your AIDS virus, human immunodeficiency virus. Okay? So these viruses affect and attack different things. Okay? The herpes virus attacks your nervous system. That's where it lives. We'll talk about the different types of herpes viruses. The AIDS virus attacks your immune system. It makes your immune system doesn't recognize things that are foreign anymore, and you can't fight off infections anymore. You become immunocompromised. Okay? Because your immune system basically circulates through your whole body. Okay? It circulates through your body and then identifies when you're developing in the fetus the different areas that are yours. It binds to your cells so the immune system knows what not to destroy. Okay? The immune system is never exposed to your nervous system. The nervous system is enclosed in the spinal column, okay? And it is separate from the rest of your body. So your immune system never gets exposed to it. So what the herpes virus does, which makes it very unique, the herpes virus lives in your nervous system. That's where your body never finds it. If you get herpes as a 5-year-old on the playground because you put in your mouth the same toy that another kid had, you're going to have it for life because it lives in your nervous system. And in your nervous system, you don't have exposure from the immune system. Okay? Oops. Let me put that off. Okay, and that makes sense. So now when you have herpes virus, get rid of that.
That's
So
so
it's living in your
cool.
immune system. What's that?
That's very interesting how the herpes virus lives in your nervous system, so you have it for life because it's never exposed to your immune system. That's crazy.
Right. And that's why if you ever get cold sores, I get cold sores on the back of my tongue and they hurt like crazy. They always come in the same place because it follows your nerve tract out and then it releases out through the system and your body can find it and then destroy it takes about 7 to 14 days for herpes to go away. And that's like your cold sores out of your lips, or I get mine like on the base of my tongue. Now, it's kind of weird because 4 things tend to make those come out. You have stress, chocolate, peanuts, and sunlight. And in the desert, there's no sunlight. And as a student, you never have stress. So those 4 things tend to bring the virus out. Don't know why, but those are the ones that usually trigger your cold sores. Now, with AIDS decreases your immune system so the herpes virus can run rampant. And they can get some very nasty infections. I mean, with these herpes viruses, to where they can't eat, they can't function. So what we do is we use laser treatments, and actually dental lasers are really effective against the herpes virus, which is really cool. And they don't cause any discomfort. My assistant, how old was Caitlin? She was probably 3 years old. She had primary herpes all over her mouth. She got them at the school. You got primary is a big case, and then this when it comes secondary, it comes out smaller each time. So the primary is very painful. So we have a laser at the office, and you just take it and you don't hit it. You hold it above, about a millimeter above, and you draw circles for about a minute. And it wipes out all the pain from the herpes, from the virus. It's really slick. It's a really neat treatment for it. So it's kind of cool how these viruses, certain viruses live in different areas, and we can't find them. So remember, naked viruses have no envelope. The helical one is the pox viruses, the icosahedral ones, the polio, and the papilloma virus. Okay. Now, how would you describe this virus? Now, the answer in the book, I think, is wrong. And I'll tell you why in a minute. Does this virus have an envelope?
Yes.
Yeah, it says it right there, right? So it cannot be naked, cannot be naked, cannot be naked, because it has an envelope. Okay?
I
Is it
think.
helical?
I
Huh?
think oh.
But go ahead.
I'm sorry. Go ahead. I don't even know what I was gonna say.
That's all right. That's no problem. I mean, is it helical?
No.
Not really. Okay. Is it isohedrical? it doesn't have that real pattern. It's all not solid. So I'd say complex and enveloped, right?
Yeah.
Okay. The book says it's helical and naked. Now let's go back. Helical means it goes like this in a rod shape. Naked means it has no envelope. It's wrong on both counts. Unless I'm missing something.
No, I agree. I think it's complex enveloped.
Yeah, I mean, it says it's envelope right there. I mean, you can't miss that part. So just if you're reading through the book, just remember this one's marked wrong. This will be marked as F. Complex and envelope. You both see why?
Maybe because the complex, like they usually have a tail or something.
Yeah, complex. Some of the complex ones can have a tail. But I mean, I can't see it being naked. I mean, it has an envelope. I mean, I would probably accept icosahedral and envelope because it is kind of shaped like polygons put together, but not as much as I'd like. But you can make an argument for that. But you cannot make an argument for helical, which is rod shaped, and naked, which is no envelope. You can't make an argument to defend A. You can make an argument to defend E. I think F is a better answer, but I'd give you credit if you put E if you argued with me. Does that make sense?
Yes.
All right. Okay, so it would be F, complex and envelope. All right, so the viral genome, remember the genome is your DNA and RNA. So it's either, we've said this probably 6 times now, it's DNA or RNA, never both. The virus either has DNA in it or RNA. It carries the genes, either one, necessary to invade a host cell and then take over the existing DNA of the cell and redirect the activity of the cell to make new viruses. So it takes all the machinery, of the cell, takes its DNA and goes, okay, I'm taking over and we're going to make enzymes to replicate our new virus. And we're going to forget everything else. So it redirects it. So the number of genes varies from each type of virus. You can have a few genes on your RNA to hundreds of genes on your RNA or DNA. So the DNA viruses, DNA is usually double-stranded, okay, but it can be single-stranded in viruses. And it's either circular or it's linear in its shape. Okay? RNA viruses, RNA is usually single-stranded, like normal, but it may be double-stranded in the virus. Okay? And it may be sent into separate RNA pieces. You may have 2 or 3 RNA pieces inside the virus. So single-stranded RNA is most common. Ready for immediate transfer are called positive sense RNA. Single-stranded RNA that must be converted into the proper form are called negative sense RNA because it has to go through an extra step to convert it to take over the cell. Okay? So there's 2 different types of single-stranded RNA. One that's called positive sense, and one that's called negative sense, which converts, replicates the cell and converts it into the one that's ready to go to work. So you have positive sense RNA and negative sense RNA for single-stranded RNA viruses. Okay? You have preformed viruses required for viral replication, preformed enzymes. Okay? You need these enzymes for our viral replication, polymerases, which cells have, because that's what synthesizes our RNA and DNA. Replicases copy RNA, which our cells do, and reverse transcriptase. That's for the synthesis of DNA from the RNA HIV virus. So it's reverse transcripted, which we do have in our cells also, that this RNA injection from the virus, from the HIV virus, we need those enzymes to replicate the virus. Those 3 are needed. How we classify viruses. Okay? The main criteria are their structure, chemical composition, and genetic makeup. Okay? Where there's RNA, DNA, single-stranded, double-stranded. Okay? There's families of viruses, which are italicized just like regular, given the substance Viridae, and genera. The genera of it is also an italicized and ends in the word virus when we label these things. Okay, so viral species, distinct virus types that share a collection of properties like a host range, pathogenicity, they infect similar cells, or their makeup is genetically the same. We group those into a viral species. For placement into a virus family, we include the type of capsid, we include the microscopic appearance, the anatomy or geographic areas, and the effects it has on the host, what it does. So the type of capsid, the nucleic acid strand number, presence and type of the envelope, overall virus size, and area of the host cells in which the virus multiplies. Okay, all those are considered in it. The microscopic appearance, okay, like a rabbit virus, what it looks like under a microscope. The areas, like an adenovirus, okay, and the hantaviruses affect certain areas. Okay. So acronyms blending several characteristics. This was called a PICO, okay, a picornavirus. Blends several characteristics together. Okay. So to summarize certain diseases with the viruses, these are DNA viruses. Okay. The variola virus, okay, is smallpox, cowpox. The herpes simplex virus. These are your fever blisters or cold sores. Herpes simplex virus 2 is your genital herpes. Okay, so the different viruses that from the mouth. But again, they live in the nervous system and cannot be found by your immune system. This is the varicella zoster virus, responsible for chickenpox and shingles. The human cytomegalovirus is cytomegalovirus infections. The human adenovirus is adenovirus infections. The papilloma, human papillomavirus, some types of papilloma warts on the body. Okay, the JC virus is progressive multifocal leukinoencephalopathy viruses effects. Okay. Hepatitis B, serum hepatitis released in the system. Okay. The parvovirus, okay, erythema infectiosum. So if you look at different DNA viruses, they come in a different shape. This is the herpes, type 1, type 2 virus, what it looks like. Okay. This is a pox viridae, chicken pox, etc. The adenovirus, papillomavirus, and the hepatinovirus. You see all the different shapes of these things that allows you to classify the different types of viruses. Poliovirus, poliomyelitis. Okay. The hand, foot, and mouth disease, coxsackie-virus. Hepatitis A, okay. Short-term hepatitis. Okay. The rhinovirus, okay. Common cold, bronchitis. Okay. The Norwalk virus is viral diarrhea. Different viruses continued here. You could have like yellow fever virus, St. Louis encephalitis virus. These are responsible for dysentery and different encephalitises, yellow fever, etc. Rubella virus is for the measles, German measles. Dengue fever virus, dengue fever. West Nile virus causes West Nile fever. Zika virus causes Zika fever. Bunya amarela virus, it's a California encephalitis. Got Cinnober virus, respiratory syndrome, and Rift Valley fever virus causes Rift Valley fever. So a lot of the names of viruses tell you the diseases that they cause once they're identified. Okay. You can have hemorrhagic fever from the Crimean Congo, hemorrhagic virus. Ebola. Ebola is a very nasty virus, causes Ebola fever. Okay. Ebola is an extremely fatal virus, causing mass dehydration of the body. Very easy to transmit. It's a shepherd's hook. It's the virus that looks like that. It's like a shepherd's hook. Okay. And there was a big scare of that about 4 years ago, as people came back with from the African countries. Colorado tick virus, Colorado tick fever, human rotavirus causes rotavirus gastroenteritis, influenza, influenza virus, Asian, Hong Kong, swine influenza. These are all your influenza viruses. Your common flu. This is what the flu vaccine helps protect against. The parainfluenza virus causes parainfluenza. Mumps virus causes the mumps. Measles virus causes measles. Respiratory syncytial virus causes a common cold syndrome. Yeah, different viruses. You see the different shapes, different rod shapes, different spherical shapes. Some have envelopes, some don't have envelopes. Some have the polygon shape. Those are your RNA viruses. So general phases in antivirus multiplication, how they multiply against themselves. The first thing a virus has to do is absorption. It has to bind to the host cell. Okay. It has to be floating through the extracellular fluid, and it has to find a host cell to bind to. That's the first step. After that, it has to penetrate the host cell. The genome, the RNA, the DNA, has to enter the host. Has to uncoding, which means the nucleic acid, remember, surrounded by that capsid, that coat, it has to be released from that capsid so it can act. Remember that capsid protects it. So now it opens, once it's in the cell, it opens it up so it can release it. Then we get synthesis. It takes over the DNA of the existing host cell and it starts to make viral components. It changes the DNA genes to make the pieces that are needed to put the virus together. Once it makes the pieces, it has to assemble them. So it has to assemble a new virus, and once all these viruses are assembled, it has to release them. It has to spit it back out so the viruses can infect. Okay? The assembled viruses are released by either two ways, by budding or by cell lysis. Budding means part of the membrane kind of expands and it makes a little bud out here, and that RNA is inside of it, and then it seals it off and releases this into the outside with the viral particles inside. Okay, or cell lysis means the whole cell blows up and all the viruses inside the cell are now released. So it either packages it and excretes it, or the whole cell blows up. So yeah, these are the main points are absorption, penetration, uncoding, synthesis, assembly, and release. That's the order it has to occur in for a virus to be effective. And here's a diagram of it. You can see what it does. This is number one. These are little spikes. Remember the spikes on the outside of the envelope or the capsule? They bind to the cell membrane. They find receptors on that cell membrane that are specific for that spike, and it allows the cell to bind to it. That's absorption. The second step is penetration. You see it comes into the cell here, starts to invade the membrane, and it gets incorporated into the cell, into the cytoplasm. That is penetration. Then uncoding, the envelope opens up so you can release the RNA. Okay. Okay, after the encoding, the RNA has to function and it starts to code the cell to make its different proteins needed. Okay. So you get synthesis of protein production. Okay. After all the proteins are made, then it starts to make the viral parts. And then you got to put these viral parts together. We have to assemble them. So once we code for it, we make all these new RNAs, goes to the ribosomes, all these new parts, and we get these new spikes, okay, that are made. And then everything is assembled into a virus, and it either buds off, like in this case, for release, or all these viruses are released when the cell explodes with cell lysis. Okay. So breaking down each section, absorption. Okay. Virus constantly collides with the whole cell. They bump into each other. Okay. They have specific proteins that bind to the receptor site, so it sticks to the cell. It sticks to its membrane. So a spectrum of cells virus can infect a whole range of cells. A lot of cells are specific to certain areas. Hepatitis B virus is specific for human liver cells. Those are the cells that it binds to and attacks. Poliovirus. Poliovirus affects your nerve cells, shuts down your respiration. That's why polio victims in the past are on respirators. Rabies. Rabies attack a whole bunch of your cells. That's why rabies is so deadly. That's why animals have to have their rabies vaccination so they don't transmit it. And so that's absorption. You can see where these spikes fit into these receptors. Okay. The spikes here bind to the receptors and it sticks to the cell. And now you have a way of transmitting the virus into the cell. Penetration, okay, and then uncoding. All animal viruses must penetrate the cell membrane, right? All our cells have cell membranes, no exceptions. Okay, so the animal virus must penetrate the cell membrane. And then once it penetrates the cell membrane, it has to deliver the viral nucleic acid into the interior of the cell. So most viruses are going to enter in one of two ways. They're going to enter by fusion or endocytosis. Fusion is the viral embryolate fuses directly with the membrane, and you rearrange the lipid so it dissolves right through the membrane. Endocytosis is when the cell membrane comes up and encapsulates the virus and brings it in. So the entire virus is engulfed and then enclosed in a vacuole or a vesicle that we've produced for the cell membrane. Once inside, the virus is uncoated, which means it releases protective coating, the capsid layer, and now you got your viral nucleocapsid or nucleic acid is now released. So then it releases your RNA or your DNA into the cell, and these take over your machinery for the cell, making proteins for your virus now. Okay, so you can see the viral spikes attack. Okay, the virus fuses with the cell membrane and injects its RNA or DNA into the cell. Okay, so that is fusion. The other one is encapsulation or endocytosis, same thing. We'll keep it the same words, endocytosis. Okay, the virus attaches to the receptors. The cell wall actually invaginates around and completely encloses that, puts a vesicle into the cell. Now this cell wall, this lining, the capsid, starts to deteriorate, and then this RNA virus is released freely into the cell, and you got free DNA or free RNA. So they both end up the same. They both end up with free RNA or free DNA. And it could be by fusion or by endocytosis. There's two different methods of penetration once the virus is recognized and binds to the receptors. Okay. You can see it again, the nucleic acid here. These are receptors, they bind to it. Okay. The cell is going to come up and engulf that and break it into the cell. That's endocytosis. Then this viral RNA breaks through and is released as free RNA into the cell to do its thing. All right, replication and protein production depend on different things. Okay, it depends whether it's DNA or RNA. DNA viruses generally are replicated and assembled in the nucleus. So if it's a DNA virus, the DNA goes into the nucleus where our DNA is and starts to assemble viruses in the nucleus. RNA viruses generally are replicated and assembled in the cytoplasm because that's where our RNA is. That's where it does our work. Remember, DNA goes to messenger RNA, which goes to cytoplasm, which translates it from the DNA, the messenger RNA and transfer RNA to staple together amino acids. So our RNA works in the cytoplasm, our DNA works in the nucleus. Same with the virus. It knows where a DNA virus is going to look for DNA. It's going to the nucleus. An RNA virus is going to look for RNA, which is in the cytoplasm. Remember, we talked about positive sense RNA and negative sense RNA. Positive sense RNA are ready to go. Okay, they contain the message they want to make their virus proteins, and they just start kicking it in. Negative sense RNA have to be converted into positive sense RNA. So there's an extra step with negative sense RNA viruses. Then you got to assemble them. Once you get all these proteins made, you got to build them. You got to put the wheels on, you got to put the doors on, you got to put the roof on, the windows, you got to build this thing. It's a functional virus, just like an assembly line for a car. So mature virus particles are constructed from the growing pool of all these parts we're making. So the capsid is first laid down as an empty shell, and that's going to serve as a receptacle for the nucleic acid strand that we're going to put in it from the virus. Electron micrographs from the taken during this time show cells with masses of virus, often enclosed in packets. So these packets are being made. So you look at all these little packets here, the viruses are inside all these little packets are going to be released. Remember, the capsid surrounds it, so it's released into the extracellular fluid or the lumen. It's going to protect that virus so it can infect another cell. and cause a huge infection. Assembled virus leave by one of two ways. We talked about that. We talked about budding or exocytosis, and we talked about lysis. Budding is when part of the membrane, you have your membrane here, this is your virus here, it fuses with it. And then it starts to open. It starts with this. I'm going wrong. Let's hold a second. Okay. It starts to fuse to it and starts to bud off. And this little piece breaks off. And that's budding. Lysis, virus particles, and this whole thing just blows up. And all these virus particles are released. Okay. So when lysis occurs, the cell dies, obviously, because it ruptures. Duration releases an envelope virus. You can see here's your virus that's been made with a symbol. This is your nucleic acid. Here's your virus proteins that have been assembled, your caspin. And it gets encoded into the budding of it, and then it gets released. And this little guy can go out and infect other cells. And this is a scan under a microscope of what we look like. And the virus is being released from the cell. Okay. Viruses commonly contain both DNA and RNA. Everybody should get this one. Oh, it can only contain one, so don't miss on the midterm. It can only contain DNA or RNA. Damage to host cells, cytopathic effects. Cells damage altering microscopic appearance. It changes the shape of the cell. We talked about it a little bit when they're encapsulated inside the cell. You get disorientation of individual cells. You get gross changes in shape and size because you're building up. You're taking this virus, you're putting hundreds and thousands of them inside the cell. It's going to change its shape. It's going to alter that cell. It has to. Intracellular changes, you get inclusion bodies and syntium. Syntium means multiple nuclei. You see in these cells, you can see 4 or 5 nuclei here, 3 nuclei here, 4 nuclei here. Okay, so you get inclusion bodies, which are these basic guys right here. They just body the viruses in them. And syntium means multiple nuclei in the cells. And you get those changes that we see. Smallpox virus. Okay? The cells become rounded. The inclusions appear in the cytoplasm. So this is how we can look at things and we can tell what virus is infecting our patients and what kind of treatment we need to do for that. Okay, herpes virus, herpes simplex. These cells fuse to form multinucleated syntia in the cell. You get nuclear inclusions. The adenovirus causes a clumping of cells and nuclear inclusions. Poliovirus causes cell lysis. The cells just start to blow up. Okay. Reovirus, the cells become enlarged. They become have vacuoles and inclusions in the cytoplasm. So these vacuoles become bigger, these inclusions build up in the cytoplasm, and the cell becomes tremendously larger. Influenza, the cells round up, and there's no inclusions. Rabies, there's no change in cell shape, but they have cytoplasmic inclusions called Negri bodies. Negri means black in Latin. So you get these black bodies building up in the cell. The measles, okay, form syntiform, multinucleate. Okay. Syntia means multinucleus. So we can look at these different effects on the cell, and we can say what type of virus you have. Because you're not going to be able to see viruses all, because you can't see viruses. They're tiny, tiny guys. Okay, but you can see the effects. And if you see the effects, then we can make a really good educated guess what virus is affecting that patient. You can get persistent infections with viruses. Okay, the cells harbor the virus is not immediately lysed. The cell doesn't destroy itself. It can last weeks or it can last the host lifetime. We talked about that. We talked about with the herpes simplex virus. It lasts your whole life. Once you get it, and 95% of kids get it, okay, once you get it in your preschool years, you know, because you're putting something in your mouth and another kid put in their mouth, which they all do. Okay, once you get it, you got it for life. It's not going anywhere. Okay. Several can periodically reactivate, which is a chronic latent state, herpes virus. It's a chronic latent state, it lives in your nerve tracts, so it's not exposed to your immune system. It just sits there, and certain things can activate it. Like I said, with herpes virus, it's chocolate, peanuts, stress, and sunlight. Those 4 things tend to bring out the herpes virus. Ever notice during exam times, if you get cold sores, that's when you get cold sores. Midterms, finals, that's when you break out in cold sores. That's when you're stressed. That virus is activated from your nervous system. The measles virus, okay, may remain hidden in brain cells for years before it comes out. Herpes simplex virus, we talked about, cold sores. The zoster virus, shingles, can come out when you're 56 years old. You get that band around your stomach or your legs or your back, wherever it comes out, wherever it's hiding in your nerve tract, it follows that track, that nerve. Remember the different nerves in your body, the different areas. It follows that nerve track and comes out to that area. Okay? So these herpes zoster virus, herpes simplex virus, the measles virus, these can all live in your body for years. Like the herpes simplex virus, this is permanent. That's not going anywhere your whole life. What damage from viruses? Some animal viruses enter the host cell and permanently alter its genetic material, resulting in cancer. So some viruses cause cancer. Remember, cancer is a growing disease. If the virus comes in and it gets incorporated in DNA and it takes over permanently, and it tells that cell to grow and make more viruses, whatever it is, and that cell starts to grow. That's a permanent change. And cancer is a disease where cells multiply or grow extremely fast. That's what causes a tumor, the rapid growth of cells. That's why we can't, the big and the devastating effects of cancer is that we can't recognize it. Cancer, this virus gets into your cell, right? It starts to grow. Does it change the outside of your cell, the receptors or anything like that? No, it doesn't change the receptors on the cell. It just grows inside the cell, makes the cell grow faster, so it multiplies. So you get more, quote, normal cells. They're not normal, but they're not changing that we can find. And they keep growing, and that's what forms tumors. Tumors that we can't identify as foreign, because these markers, the immune system is recognizing as your own body. We've already recognized it, it's your own cell. So we can't recognize it as foreign, so we can't defeat it. That's what makes cancer so deadly. It's our own cells where there's no markers to identify it as bad. Now, with saying that, about in the last 20, 30 years, immunologists have identified something called NK cells. NK cells stand for natural killer cells. This is part of our immune system, and it goes through our body, and it can identify a cancer cell. We don't know how, we don't know why, but it can identify a cell with the same receptors on it, but just has a virus growing inside of it, and it kills the cells. That's why some people get cancer lesions, some people don't. These NK cells seem to be a key in doing that. There's not a lot known about it yet, a lot of research being done, but there is a way they're identifying cancer cells, and we don't know how they're doing it. Because the cell is not changing its outer receptors. Okay? So the viruses that cause this are called oncogenic, and their effect is called transformation on the cell. Transformed cells have an increased rate of growth. Cancer cells grow faster. That's what causes tumors. There's alterations in chromosomes and the capacity to divide for indefinite time periods, resulting in tumors. It makes our own cells grow faster. Viruses in mammals initiating tumors are called oncoviruses. The papillomavirus is cervical cancer. That's what it causes. The Epstein-Barr virus causes Burkitt's lymphoma. Okay? It's kind of neat. These guys are really interesting. Okay, multiplication in bacteriophage. The bacteriophage, remember we talked about bacteriophages. Bacteriophages are usually called phages, and all they are is viruses that infect bacteria. They don't infect human cells, they attack to the cell wall. So you see bacteriophage, all it is a virus that affects or attacks bacteria. And we can study those. We can study those. Bacteria have such a rapid generation rate, rapid turn rate. Every 10 minutes, bacteria can replicate. We can see the changes, and we can use that to study bacteriophages, and we can apply that to viruses that affect humans. So the most widely studied are E. coli viruses. Their structure, and E. coli is a DNA virus. Multiplication goes through similar stages as animal viruses. It goes through mitosis. Remember the same prophase, metaphase, anaphase, telophase, and then interphase resting cycle. Okay? The virus goes through those same stages extremely quickly. Our generations take roughly, depending how old you want to be, roughly 20 years for us to replicate a generation. Virus can go every 10 minutes. So we can see genetic change in viruses much more quickly. And that's when we study them. Okay. So only nucleic acid enters the cytoplasm. Okay. So uncoating is not necessary with phages, because with phages, it's a funky looking virus, drills through the cell wall, and it injects the raw RNA or the raw DNA in this case directly into the cell. So it does not have an uncoating phase because there's no coating around it. The coating stays outside the cell. Okay? So if you have your cell, as a cell wall, your virus comes here, and these are your phages. They look kind of funky. They have these legs on them. Virus comes up here, a little gap, and this is your DNA right here. So these attack. It has a little drill that comes down. It doesn't have a little drill, but it has enzymes and proteins that open up your cell wall. Okay? Then this, our DNA, in this case, comes through that opening and comes in here. So now you have DNA directly in the cell. That's uncoated. So you do not have to have an uncoating phase because there's nothing around it. There's nothing to uncoat. Remember we had encapsulation, we brought it in, we had to get the coating off of it, or we had the exocytosis, endocytosis that brought it in, I'm sorry. We had to uncoat it. This one you don't have to. So release is a result of cell lysis induced by the viral enzymes and accumulation of the viruses. This is what's called a lytic cycle for a phage. Gets injected, builds up more of the viral proteins, assembles them, builds up more and more and more, and the cell just kind of goes apart. And that's cell lysis, and these phages can now go onto different cells and infect those different bacteria. Okay? It's called the lytic cycle because it lyses, it blows up. All right, so step in phage replication. Okay, notice there's no uncoating phase because you don't need it. It's not being injected with a capsid around it. You had absorption, same as an animal cell. Binding the virus with specific molecules on the host cell. Bind to the outside. Once it binds outside, it drills through, penetration. Then the genome enters the cell, the DNA or the RNA. Then once it's in the cell, we get replication. Remember that this would normally be uncoating right here, but we do not need it. So replication, viral components are produced, then it assembles the virus parts, okay, like a car assembly, puts them all together. Then you get maturation. The virus starts to mature, completion of viral formation. And then once the virus is formed, you get lysis. So the cell blows up, explodes. The lytic cycle involves full completion of viral infection through lysis and release of the virons, the viral particles. Okay? That's called the lytic cycle for bacterial phages. And then it repeats itself. It infects another cell by, once the cell blows up, the viruses that are released go back to absorption. Occasionally, the virus enters a reversible state of lysogeny, and its DNA is incorporated into the host genetic material. So every once in a while, it doesn't complete the cycle, but it goes into and it goes in the DNA and becomes part of that DNA and is transferred cell to cell, depending on the virus. So here's your multiplication for viruses. Okay. So this is your lysogenic state, as it's sitting here. Then you get penetration. The virus comes onto the cell, drills through there, and it releases DNA. The DNA can come in and it can do two things. It can take over and start making a tremendous amount of virus particles, or it can go this way and incorporate it into the DNA and become part of the DNA itself. This splices with it. This is how we use phages in genetic engineering, because we can incorporate DNA strands into this circular pattern. And then it can multiply extremely quickly, and we can control now the bacteria to produce the enzyme we want. We can produce insulin that way. Okay, we can put the insulin gene inside that bacteria. Now the bacteria has a cell that says produce insulin. So it produces insulin. Okay, this is what's called genetic engineering, by using these circular DNA, these strands, to make the genes that we want. Then if the cell lysis cycle, it makes a whole bunch of particles. Then it assembles them. Then all these assembly parts get put together to form a virus, and they mature. And then the cell breaks, blows apart, and all these viruses get released. And then those go back and reinfect. Okay, and that's your lytic cycle.
So for genetic engineering, we have to use bacteriophages.
Yeah, that's what we use. We use
Yeah.
bacteria that are infected with these phages because this circular virus is very small and we can control it a lot easier. I mean, our DNA is huge.
Because we use the strands of the bacteria.
Correct. The little circular bacteria, we inject it right into that.
So how do you do that during inculation or whatever? Like you take the
Yeah, we inoculate it. We make our own viruses, our own phages, and we inject our own bacteria into it. And it gets our own DNA that we want. And it gets incorporated into the phage, bacteriophage.
so that's what makes like insulin.
Yeah, that's we use bacteria to make insulin. We make different kinds of medicine that we need to make. I'm not sure the other ones that we use for bacteriophages for, but yeah, that's all our genetic engineering.
That's
Kind of interesting. Huh?
yeah, really cool.
Yeah, it's neat how they do that. I'll look up more of the things. I know we make insulin that way. I'm not sure of other products. I'm drawing a blank on this right now, but I'll look some up. But this is the bacteriophage. This is what they look like. This is the head. Inside the head is our genetic material, or DNA or RNA. So these little guys here, these little legs basically attached to the cell. This has little coils here, and this drills down and forms an opening through the cell wall. Bacteria have a cell wall, we don't. Okay? And then this DNA comes out as it injected through the tunnel, through the tube, and comes out directly into the cell. That way it doesn't need to encode because nothing's around it now. It injects our pure nucleic acid into the cell, into the cytoplasm. And this is the lysis. This is the cell right here. This is your cell wall. You can see it's been broken apart. So these are all virus particles, and it blew apart a hole right in the cell here. So all these virus particles are being released into your bloodstream, into your intestines, wherever the lumen is that you're releasing into. So this is a difference between the different procedures that we just gone over, like absorption. We get precise attachment of special tail fibers to the cell wall. This attaches a capsid or envelope to the cell receptors. So with penetration of that, we inject nucleic acid through the cell wall for bacteriophage. This goes through the cell membrane and it incorporates into it, okay? So we have to have release of that capsid uncoating for it to be active. Okay? You get synthesis and assembly, it's pretty close to the same. Okay, but remember bacteria have no nucleus. So it's all factions in the cytoplasm. So DNA viruses affect the nucleus here. DNA viruses just replicate in the cytoplasm here. For bacteriophage, you get lysogeny, which means the cell blows apart. Or in animal cells, it can go with budding off. It doesn't have to break the whole cell apart. Okay, lysogeny means it incorporates it into it. Okay, cell lysis after being weakened by cell virus. So all bacteriophages break the cell apart unless it goes into lysogeny, then it stays. Okay, some cells lyse or some uses budding, sudden lysis or slow budding, or sudden lysis, breaking apart of the cell. So there are a lot of similar parts, a lot of different phases they undergo. Okay, so not all phages complete the lytic cycle, which you talked about, like when we're doing genetic engineering. So some DNA phages called temperate phages undergo absorption and penetration, but do not replicate. Okay, so the viral genome inserts into the bacterial genome, just like we do when we're supposed to enter its DNA, become an inactive phage, a prophage. So the cell is not lyse, cell doesn't blow up. It incorporates that DNA into the bacteria, and that bacteria is now replicated, and it has that DNA over and over and over again. So the prophage is retained and copied during normal cell division. Okay. Results in a transfer of the temperate phage of that genome, that nucleic acid, to all host cells progeny. And that's lysogeny. It doesn't cause lysis, but it's incorporated gene over and over every time it replicates. So induction can occur, resulting in activation of lysogenic prophage, followed by viral replication and cell lysis. So once it becomes activated, then it can make more phage viruses and activate, then break apart the cell. And so it can live there for a long time. Lysogeny results spread of the virus without killing the host cell. Okay. So phage genes in bacterial chromosome can cause the production of toxins or enzymes that cause pathology, lytic conversion, bacteria, diphtheria, cholera, botulism. All that can be caused by the gene coming into there and causing the production of endotoxins, enzymes that cause pathology and not rupturing the cell itself. Okay, which of the following is a step found in animal viruses, multiplication, but not in bacteriophage replication? So what happens in animals, but not in bacteria?
Is it D, assembly?
Because they all get absorbed, right? They all penetrate. The viruses get built in all of them, and they all get released. Remember, the bacteriophage, it injects the pure RNA or DNA directly into the cytoplasm. So there is no encoding. Animal viruses are incorporated with the membrane around it with a capsid. Those have to uncoat and release the RNA or DNA into the cytoplasm. So with a bacteriophage, you have no uncoating, because remember, it's that you get your cell membrane here and you get your bacteriophage. I'm not good at drawing, but here's your bacteriophage. And it drills a hole through the cell wall. And now your DNA or RNA gets deposited directly into the cell. So there's no uncoating. This is not coated with anything. Okay, just raw DNA. With the regular one, with our cell, it binds to it with certain receptors. This bacteria binds to it, and the cell starts to grow around it, incorporates this whole thing inside of our RNA, and this has to uncoat to release the RNA or DNA into the cell. Does that make sense?
Yes.
Yeah, I know you can tell by my great diagrams.
Yeah, there's something special.
Yeah, no, my son's the artist. He Nitro Circus. He does all the design. He's a graphic design artist. His drawings are unbelievable. Mine are terrible, but I usually get the point across. Uncoding is the difference between animal and bacteriophage infections. So cultivate, identify animal viruses. The primary goal of viral cultivation, okay? We want to isolate and identify viruses in a clinical specimens. We want to prepare the virus for vaccines, like we talked about. You take the RNA, the DNA out, deactivate it, and use it for our vaccines. We allow detailed research on viral structures, multiplication cycles, genetics, and effects on our host cells. We can study all that with viruses. In vitro, we grow the viruses on tissues in a culture. So cell support viral replication and preservation. In vivo means inside a living, inside, not in a culture. Okay, we use bird embryos, intact self-supporting units, complete with its own sterile environment and nourishment with embryonic tissues that support viral multiplication. That's where we grow our vaccines for our that's where we grow our DNA, our coatings that we need for the vaccines is inside of egg embryos. So that's why you can't, if you want to, people that are allergic to eggs cannot get the vaccination for viruses because it's grown in egg out in the eggs. Okay. Live animal inoculation we can use. Animals exposed to injection of viral preparation into the brain, the body, muscles, body cavity, or foot pads. Growing in a dish in vitro, you can see the different areas of the culture growing. Okay, or in the bird embryos, this is where we inject it in. We inject it into the anion, or okay, inoculation of the chorionic membrane or into the yolk sac. We allow the viruses to grow inside of here, being protected and nourished by the egg embryo. Viruses are the most common cause of acute infections. There's several billion viral infections every year. Some viruses have very high mortality rates. Ebola virus, one of the mortality rates. Possible connection of virus to chronic afflictions of unknown causes. It can come over and over. It could be a virus that lives in your nervous system. Viruses are major participants in our Earth's ecosystem. Viruses are tremendously important. Detection and treatment of viruses are more difficult than other agents. Bacteria we can isolate, we can grow, we can find out what they are. Viruses are too tiny to see. Okay? We consider their overall clinical picture. We take appropriate samples. We infect cell cultures to look at those, look for how they affect the cell. We screen for parts of the viruses that we can identify, and we screen for immune responses or antibodies that are made. Like in the flu vaccine, we make antibodies to it. Antiviral drugs can cause serious side effects. A lot of these anti-cancer drugs are chemotherapy. And as you know, chemotherapy can have some very devastating side effects. So antiviral drugs are not easily tolerated by a lot of people. Non-viral infectious particles. These are prions. Prions are an interesting group all to themselves. They have some very, very special properties. Okay? Their prions are misfolded proteins, and they contain no nucleic acid, but they can solve some really bad diseases, really fatal diseases. Okay? Extremely resistant to usual sterilization techniques because they got nothing to kill. There's no nucleic acid in there either. Okay? They cause transmissible spongiform encephalopathies, which is a fatal neurodegenerative disease. It affects your brain tissue. It's a neurodegenerative and it isn't fatal. It's like a very fast-paced Alzheimer's. Okay? It's common in animals. It's called scrapie in sheep or goats. It's called bovine spongiform encephalitis, called mad cow disease. Okay? It's a wasting disease in elk, and in humans, Creutzfeldt-Jakob syndrome is called by prions, and that's a fatal disease of the brain.
So it's not like Lou Gehrig's disease.
No, Lou Gehrig's disease is a virus.
Did you say it's a virus?
Yeah, Lou Gehrig's disease is a virus. This is Creutzfeldt-Jakob syndrome, is one that's caused by prions.
Oh, okay.
Yeah, and these prions, this is what it looks like on an X-ray picture here. But they attach to the cell, they come into the cell, and they start lining themselves up. And they convert other proteins into prions, which is really strange. And then these prions build in the brain tissues, and they build up and they start to accumulate in these bands, and these bands start to build up and these huge chains, and it stops the brain function. It blocks all your nerve functions. That's how it works. I thought there was more on prions. Okay, but this is, I mean, it's these very small proteins that come, how they're transmitted. Okay? They're transmitted by eating infected meat with prions. They're very common in cannibalistic countries where they eat the human brain. Okay? If you eat the human brain, most people in our country don't eat human brains. So we don't see a lot of these diseases. But you can get it from mad cow disease. Okay? We're bovines because you do eat cows. That's where there's a big push. They have mad cow disease, they shut everything down and they isolate it. They stop that meat from going to market because of these prions. And that's how they work.
Transmissible through meats or eating human brain?
Eating infected tissue with other prions, yeah. It's
That's
not airborne or anything like that, but it's by eating infected tissue.
Okay.
Okay. Satellite viruses depend on other viruses for replication. In other words, not only does the virus depend on the cell, you got to have another virus present. It's called satellite viruses. So the adeno-associated viruses, they replicate only in cells that are infected with the adenovirus. So if the adeno-associated virus comes to a cell, it can't do anything. If the cell already has an infection with the adenovirus, then the adeno-associated viruses can all start to replicate. So you have to have a virus infecting first before these viruses can infect. These are called satellite viruses. A delta agent, a naked strand of RNA is expressed only in the presence of hepatitis B. So only in cells that are affected by hepatitis B will this RNA, this delta agent, express itself. Otherwise, it won't cause any effect. Okay. We also have things called viroids, virus-like. They're short pieces of RNA, they have no protein around them, and you only find it in plants so far. All right, so exposure to nucleases that degrade DNA and RNA would damage all of the following except what? What would, if you play nucleases destroy DNA and RNA, they degraded. So which one of these would not be affected by nucleases?
Prions?
D.
Prions, right? Prions have no DNA, RNA. Animal viruses have DNA, bacteria have DNA, cellulite virus have DNA, viruses have DNA. They all have strands of nucleic acid. Prions are only protein. There are no nucleic acid in them. Remember, these short proteins that convert other proteins to be like it. So it has to be prions. Because there's no DNA or RNA in a prion. It's only a protein. Every one of the other ones has nucleic acids in them.